Virus multiplication. Let us start with bacteriophage. Bacteriophage multiplication consists of two different types of cycles. The first one is called the lytic cycle. The phage causes lysis and death of the host cell. Lysogenic cycle. The viral genome integrates into the host chromosome. So the prophage DNA is incorporated into the host DNA. When a bacteriophage goes through the lysogenic cycle, the results of lysogeny can be two outcomes. Phage immunity. The host is immune to a phage of the same type. So if I was infected with phage A, I am now immune to phage A. However, if phage C came along, I am no longer immune to phage C, so it can affect me. Phage conversion is when the host receives new properties, and we see this in Clostridium diphtheria. The lytic cycle. The lytic cycle could be remembered by this mnemonic, APBMD, an all points bulletin for a doctor. APBMD, A stands for attachment, P stands for penetration, B stands for biosynthesis, M stands for maturation, and D stands for dissemination. Attachment is when the phage attaches by its tail fibers to the host cell. So if we take a look at this picture, here's our bacteriophage. If you remember, the head is what shape? If you said polyhedral, that is correct. And the tail sheet is what shape? If you said helical, you are correct. So here's the bacteriophage, and it's going to attach to the host plasma membrane. So remember, it's going to infect what type of cells only? If you said bacterial cells, you are correct. So here you'll see that we have our cell wall and a plasma membrane. So the virus is going to attach to the host plasma membrane through the cell wall, and it attaches by using these tail fibers. The second step is penetration. Phage lysozyme opens the cell wall. The tail sheet contracts to force the tail core and the DNA into the cell. So after attachment, the next step is penetration. And what ends up happening is, do you see the helical part? It looks like a pearl necklace, right? That pearl necklace is going to contract. When the pearl necklace contracts, that forces the tail core. You see here? Here's the tail core. It gets forced in, and it injects the DNA into the cell. The next step is biosynthesis. Biosynthesis is the production of the phage DNA and proteins. So it's going to cause that bacteria to produce more DNA and proteins. The next step is maturation. It's assembly of phage particles. So it's kind of like an assembly line. Once we made all the parts, we now need to put them together. So that's what this picture is showing you here. We have the proteins and we have the DNA. So we're going to go ahead and put it all together and make another bacteriophage. And then finally, dissemination, which is release. And it's usually released by lysing the cell. Phage lysozyme is going to break the cell wall. So this virus is going to tell the bacteria, go ahead and make lysozyme. And when that enzyme is made, it's going to be, or when it's expressed during maturation, it causes the cell wall to break open and hence lysing the cell. So now let's take a look at a viral one-step growth curve. We know that this is the number of days over the number of virions that we have released. And what you notice is that the number of virions are going to decrease. Why is that? Why does the number of virions decrease? Well, the number of virions decreases because the virus inside of the bacterial cells are inside of the cell and we can't see them. But however, after it goes through attachment, penetration, maturation, right, Then, when the virions are released into dissemination, now we can see the number of virions being released. And you see that number goes very high. And then, of course, if the number begins to go down again, why? Because they're now entering new host cells. Okay? The lysogenic cycle starts off like the lytic cycle with attachment and penetration. However, there's a different step that gets involved. So here we see attachment, then we see penetration. The next step is integration. And in integration, this is when the viral DNA becomes that prophage DNA. It gets integrated into the host. So we've injected the DNA, so now the virus is pink, the bacterial chromosome is purple. It's now going to integrate with one another. See that there? So we create this prophage DNA here. And then the cell divides like normal, replicating the prophage DNA while it reproduces. So reproduction within the host. So it's going to go through normal binary fission. Every time the bacteria goes through binary fission, it's going to have a copy of this prophage DNA each time. It remains a prophage until induction. Induction means that the lytic cycle is induced once again. And this is induced by environmental factors. So it's going through this cycle many times until there's some type of event or change in the environment that causes it to now re-enter the lytic cycle. So once it does that, we come back to the middle here. We're now going to see that it's going to go through biosynthesis. So we're going to make more DNA, more protein. Then we're going to put that together, mature, and then we're going to release the lysozyme, express it, and hence we're going to see dissemination and release. Animal virus multiplication is similar to bacteriophage, except there's one extra step. So let's go through this. Attachment. The virus attaches to the cell membrane. So you have receptor sites on the animal cell plasma membrane that may be glycoproteins or proteins. And the receptor sites vary per individual host, varies upon that individual's susceptibility. The next step is penetration. And penetration is either by endocytosis or fusion. Endocytosis is to form vesicles, and this is what we see with the naked viruses, or envelope virus, which means it has an envelope. So naked viruses do not have an envelope. An envelope virus does. So if I'm a naked virus, I'm going to come in by endocytosis. If I'm an envelope virus, I'm going to come in by fusion of the envelope to the plasma membrane. The next step is called uncoating. Uncoating is done by the viral or host enzymes. So lysosomal enzymes of the host may do this. Enzymes encoded by the virus or enzymes in the host cytoplasm will basically cause the separation of animal virus genomes from their protein coats. Okay, so the separation of the animal virus genomes from their protein coats occurs inside the cell. From there we enter biosynthesis, and it differs for DNA and RNA viruses. Of course, the biosynthesis is the production of nucleic acid and proteins. We are going to take a look at each example of the biosynthesis for a DNA virus and an RNA virus. And the examples we're going to look at is herpes viridae or herpes virus and retroviridae or HIV virus. Maturation is when the nucleic acid and the capsid proteins assemble. So you're putting the virus particles together. Dissemination or release of the new progeny. And this depends on envelope versus naked, lytic versus non-lytic. And this could be by budding. Budding occurs by envelope viruses. So budding is non-lytic. Budding is non-lytic, and it occurs within envelope viruses. And of course, lytic or naked viruses rupture. Okay, so they leave by rupture. So if I'm a naked virus, I come in by endocytosis and I lyse the cell on my way out. If I'm an envelope virus, I fuse with the plasma membrane and then I bud out at the end. So let's take a closer look at this endocytosis. So here is a naked virus coming along. It attaches to the plasma membrane, and then of course, endocytosis is creating a vesicle and bringing it in. fusion, as you can see, this guy here has an envelope. So it's going to fuse with the plasma membrane. And when it fuses with the plasma membrane, it leaves the envelope behind. Okay, so the envelope stays there as a part of the plasma membrane. Then, if I'm an envelope virus, I'm going to leave by budding. So you'll see here I have my capsids and my nucleic acid all together. I remember one of the things we learned about the chemistry of the envelope virus was that it's primarily lipids and proteins. Well, guess what? Here we see all of the lipids and proteins. And what ends up happening is going to bud out of the cell and take a piece of that plasma membrane with it. Okay? It doesn't lyse the cell at all. And you can actually see a nice picture of this occurring here. So multiplication of herpes virus. Example of a DNA virus is herpes viridae, which includes the species simplex virus. Simplex virus causes cold sores. The first step is attachment, penetration, and uncoating. Uncoating, the DNA is released into the nucleus. From there, we have early transcription and translation that makes the enzymes required for the multiplication of viral DNA. So we're going to transcribe and translate early to express DNA synthesis genes. What am I saying? We're going to make the viral DNA polymerase. Without DNA polymerase, we can't make new DNA, right? We learned this back in chapter 5. So I need to have the DNA polymerase made first. Once DNA polymerase is made, then what can I do? I can make the new DNA. So DNA replication starts. Then I can have late transcription and late translation that starts later to express the gene, the DNA that codes for the capsid and the lysozyme. So I had early transcription, early translation to make DNA polymerase. Now I have DNA polymerase to make a new DNA, and on top of that, I can also go ahead and express that DNA, make the mRNA that codes for the capsid proteins and the lysozyme protein, and translate them and make them. From there, I go through maturation, and then finally, I go through dissemination. And remember, non-envelope viruses are released from the cell by lysis. So the lysozyme is going to be expressed, and that allows them to be released. So here's a picture tying that all together for you. So you see you have attachment of the cell to the host, and if it's a non-envelope virus or a naked virus, it's going to come in through endocytosis. Once it's in, it's going to be uncoated. So do you see that? We're going to remove the capsids from around there, and we're going to have the DNA free now. That moves into the nucleus. This is the nucleus here. And the nucleus is going to then transcribe the mRNA to make DNA polymerase. So we make the DNA polymerase, of course, in the cytoplasm. Now I could come back and double up on the viral DNA as well as make the proteins, right? So make the mRNAs to make the proteins, put it all together, and then I release it from the cell. Multiplication of retrovirus. The retroviruses are RNA virus. The HIV is a retrovirus. These viruses appear to have a backward mode of nucleic acid replication. So let's take a look. So entrance into the cell. So they have attachment, penetration, and uncoating. From there, we're going to have reverse transcription. We're going to have reverse transcription of one of the two RNA genomes into a single-stranded DNA that is subsequently converted to a double-stranded DNA by reverse transcriptase. So what that means is we're going to have a single-stranded RNA. So we have a single-stranded RNA. We're going to use the enzyme reverse transcriptase. Reverse transcriptase takes that single-stranded RNA and makes a complementary strand of DNA. From there, that DNA strand is then converted into a double-stranded DNA. So we started off with an RNA. We're going backwards, backward mode of nucleic acid replication. Now, here is where HIV is the bad dog. Okay? From there, you then have the next step, which is integration. You have integration of the DNA copy into the host genome. And this is called a provirus. Do not get this confused with the bacteriophage. In the lysogenic cycle in bacteriophage, it's called prophage. When we're talking about animal viruses, it's called a provirus. So that provirus form is going to be integrated into the DNA. Okay? So the integration of the DNA copy into the host genome. So every time your cell goes through mitosis, it's making a copy of that provirus. You now can never get rid of it. Okay? And it's just basically a latent disease. And of course, your viral proteins leading to the virulence are also going to be expressed here. Then we have biosynthesis, in which you're going to have transcription of the viral DNA, leading to the formation of viral mRNAs and progeny viral RNA. So DNA is transcribed to make the RNA for the new virus, and the capsid proteins are made. Then we have maturation, and then finally, remember, dissemination. The retroviruses are envelope viruses, budding of envelope virions at the cytoplasmic membrane, and then released from the cell. So the viruses are released from the cell by budding. Okay, so budding of envelope virions at the cytoplasmic membrane and released from the cell. So let's take a closer look at how HIV works. So here you'll see that HIV is going to come into the cell by fusion because it's an envelope virus. And remember, it has 2 copies of this RNA. So it's now going to be uncoded. And that single-stranded RNA, do you see it here? It's going to be reverse transcribed due to reverse transcriptase. This is the enzyme. This enzyme is now going to lead to a complementary strand of DNA. Then that DNA is then used to make another copy of DNA. So now you have a double-stranded DNA. Now we're moving to the nucleus and integrate it into the host DNA. So see, viral DNA, host DNA. It now becomes a part of that virus DNA, part of the host DNA. It is called the provirus. And of course, it's going to stay there. So we can still go ahead and make new viruses. So we go ahead and do biosynthesis, maturation, and of course dissemination. And the only way it's going to leave is by basically budding. And it doesn't like the cell. This is table 3 on page 177 in your textbook. It ties it all together for you. Here ends part B of chapter 6.